Okay, I would like to thank you for this opportunity to talk in front of this distinguished audience about the stimulation of immunosurveillance by fasting and caloric restriction mimetics. So I'm mostly known in the Gustave Roussy Institute because I'm advocating over the last 10 years together with Laurent Sidvogel the idea that whenever anti-cancer therapies work, they do so because they are able to start an anti-cancer immune response, explaining why some treatments, unfortunately not so many, are able to achieve long-term remissions or stabilizations in patients that go beyond treatment discontinuation. So we have success stories like the treatment of, for instance, breast cancer, that nowadays in more than 85% of the cases is cured for at least 5 years. Now, I'm not so well known for a side project that we're leading that is dealing with aging. And so the hypothesis that we are advocating here is rather simple. It's the idea that any kind of pharmacological, nutritional, genetic, or behavioral manipulation that increases lifespan in model organisms, so usually we work with worms and flies and yeast and sometimes with mice, and that is also reducing aging-associated comorbidities, one of which is cancer, has to induce autophagy in many cells of the body to be efficient. And so I have to introduce the phenomenon. Autophagy is one of the most spectacular things that you can see in a microscope that our cells are doing when they become vacuolated. So portions of the cytoplasm, entire organelles, including mitochondria, or protein aggregates are isolated by phagophores that create two membrane vesicles called autophagosomes that will fuse with lysosomes to create autolysosomes. And so, in a way, autophagy is a process of self-eating in which the dents are the phagophores, the mouth is the autophagosome, the stomach, the lysosome, and the whole process leads to a sort of digestion of the luminal content. It's recycling, generation of ATP from the catabolism of macromolecules, and hence it's a catabolic process that is being done in our cells that has two consequences: an increase in energy expenditure within the cells, rejuvenation of the cytoplasm because it is destroyed and can be renewed, and adaptation to stress, especially in conditions of nutrient depletion. And as a cell biology phenomenon, it is, of course, subjected to a very complex regulation. There are hundreds, perhaps thousands of genes and proteins that regulate the process. And for this demonstration, we just need one protein to know about. This is LC3, which is usually all over the cell, diffuse, and can be followed by cell biology tricks to study its relocation to autophagosomes and autolysosomes. So we measure actually autophagy by following the redistribution of LC3 to so-called cytoplasmic dots or puncta. Now, autophagy induction by systemic means can improve many of the cellular and molecular hallmarks of aging that we defined a few years ago in a hallmark paper in Cell. So autophagy improves or reverses stem cell exhaustion, guarantees for genomic stability, improves proteostasis, can avoid deregulation of nutrient sensing. Due to the renewal function, it maintains mitochondria fit, and it also affects intercellular communication, especially the communication between immune cells and malignant cells. And so what we are trying to understand in our laboratory is the mechanisms through which autophagy induction can improve the health in model organisms. So one of the best way to induce autophagy, the physiological way that is maintained from yeast to humans, is induction of starvation, hunger, absence of calories. And this leads biochemically to the depletion of one of the central metabolites, which is acetyl-CoA, with consequent deacetylation of cellular proteins, especially in the cytoplasm, leaving the brakes from the autophagic process and hence inducing the phenomenon. And so, just as an example of the metabolic consequences measured here in the laboratory in mice that have been starved for 2 days, causing 20% of weight loss, so it's quite dramatic. Imagine this for yourselves. And then you see in the plasma, in this example, after starvation, multiple changes in hundreds of metabolites. They go up or down, so you have ketoacidosis and depletion of amino acids and glucose and all what you would expect from a biochemical textbook, and much smaller changes for the intracellular metabolome, here exemplified for circulating leukocytes. You see the changes are much smaller, they are much less significant, and exactly the same can be found in humans. So these are volunteers from our laboratory that haven't been eating anything for 4 days, so they're starved. This is myself, number 4, giving the example. And you see here these similar changes, much close to what we'll see in mice, although the weight loss is ridiculous, and also the much buffered changes in the cytoplasm of leukocytes. Now, in spite of these much smaller changes in the intracellular metabolome inflicted by starvation, we can measure autophagy. So the immunofluorescence staining of LC3 in lymphocytes or neutrophils, but also in all other major leukocyte subpopulations, will show you the appearance of these cytoplasmic LC3 positive structures that are actually autophagosomes. And as a biochemical correlate, you will find that if you measure global protein acetylation, always by immunofluorescence, among circulating leukocytes from mice or from humans, this signal in the cytoplasm goes down, indicating a decrease in acetylation of cellular proteins. Now, the biochemistry that is behind is quite simple. It's acetyl-CoA, and if we just zoom on this part of the chart, you have to know that the cytosolic pool of acetyl-CoA, that is the one that matters for the regulation of autophagy, comes from glycolysis, from amino acid catabolism, and from beta oxidation. So fats, proteins, and sugars all at the end contribute to the generation of acetyl-CoA. So any kind of calorie from that point of view is relevant. And so our hypothesis is that deacetylation facilitates autophagy, but hyperacetylation, too much acetylation, due to an excess of acetyl-CoA resulting from caloric excess, can disable autophagy and hence have a negative impact on this anti-aging mechanism. So fortunately for us, there's not only there are alternative strategies to induce autophagy that are not consisting in starvation and that consist in other ways to reduce protein acetylation. So we can reduce protein acetylation by inhibiting the synthesis of acetyl-CoA, so inhibiting the enzymes that assure the final production of acetyl-CoA. We can inhibit the proteins, the enzymes that are required to transfer the acetyl group from acetyl-CoA on proteins, so the acetyl transferases. And we can activate those enzymes that deacetylate proteins, the deacetylases. And so the bottom line is that we have different classes of compounds that we call caloric restriction mimetics that can achieve at the final outcome protein deacetylation and induction of autophagy, although their primary mechanisms are different. So For the inhibition of acetyl-CoA generation, we have, for instance, hydroxycitrate that it inhibits ATP citrate lyase, the final cytosolic enzyme that generates acetyl-CoA, or the largest pool of acetyl-CoA in our cells. We can also use chemically defined high affinity compounds generated by the pharmaceutical industry. We have inhibitors of the acetyltransferases, such as the natural compound spermidine, or the green tea flavonoid epigallocatechin gallate, or salicylate, which is a plant stress hormone and is the drug that is the effective molecule generated from the prodrug aspirin. And we have chemical inhibitors such as C464 that inhibits AP300 as well. And we can activate deacetylases such as sirtuin-1 with resveratrol contained in red wine and perhaps other compounds. And so all these caloric restriction mimetics may be interesting to explain the phenotype of modern man that had a civilized ancestor, Homo sapiens. And our idea is that there are not just there's not just the choice of deacetylation and hyperacetylation by obvious dietary choices. It's not any publicity for French products here, but that in addition you can intervene on acetylation reactions by natural compounds or chemical compounds that are the caloric restriction mimetics. And that hence can shift the balance between metabolic health and metabolic syndrome and disease by affecting the acetylation, deacetylation reactions. And so we have the example of a few compounds in our diet that actually affect the autophagic cascade through these reactions. So this is a result that has been, is a 20-year-long experiment that has been done at my alma mater, which is the University of Innsbruck, in which they followed a population of some 3,000 individuals living in a small valley in northern Italy. So these people are socioeconomically homogeneous. They don't move around in the world. They are in this valley, and they use always the same recipes for their food intake. So using food questionnaires, it was possible to calculate the precise molecular composition of their lifetime diet over 20 years for 174 macronutrients and micronutrients. And then this information was correlated with overall mortality and morbidity over 20 years. And it turns out, not very surprisingly, that trans fatty acids that are contained in industrial food and in dairy products, as well as in beef, trans fatty acids, be they polyunsaturated or monounsaturated, have a very negative impact on overall survival of the population. But there was one ingredient that turned out to have a positive impact, the most positive, which is spermidine. And actually, we are working on the mode of action of these molecules to come to the conclusion that trans fatty acids can inhibit autophagy in specific circumstances, while spermidine is an inducer of autophagy because it is a caloric restriction mimetics. And actually, spermidine in this cohort reduces mortality due to the three major causes, which are cardiovascular, as we heard this morning, and also cancer. And finally, the third group of epidemiological reasons for death in patients, which is other. Everybody, by the way, wishes to die from the other cause. We don't know why. Now, spermidine has been extensively investigated in our laboratory and others for its effects on aging and disease in mice, in rats, in flies, in yeast, in all model organisms that you can imagine. And it turns out to generally increase lifespan in an autophagy-dependent fashion, meaning that genetic inactivation of essential autophagy genes and proteins that stop the process will abolish the positive effect of spermidine on lifespan and on many of its health-improving effects. For instance, spermidine totally wipes out cardiovascular aging in mice, and it improves, as I will show in a minute, the anti-cancer immune response in model organisms as well. So when we implant a cancer in immunocompetent mice and wait the tumor to become palpable and then treat it with one shot of systemic chemotherapy, for instance, the anthracycline mitoxantrone or this platinum compound oxaliplatin, you see by comparing the black untreated line and the blue chemotherapy treated line that there is a therapeutic effect. Not fantastic, but you reduce tumor growth for several weeks after one single dose of chemotherapy. And if you give starvation as a therapeutic intervention on the 2 days that flank chemotherapeutic treatment, you see that starvation on its own, the green line, has no impact on tumor growth, but if combined with chemotherapy, there is an hyper-additive, perhaps synergistic effect that, interestingly enough, is completely depending on T lymphocytes, because if the same tumor is implanted in nude mice that lack a thymus and hence lack mature T lymphocytes in the periphery, these mice with the same treatments don't show any tumor growth reduction. So it's an immune-dependent therapeutic effect. We also know that it's an autophagy-dependent effect, because if we inactivate autophagy in the tumor cells, the whole treatment regimen doesn't work anymore. And now, fortunately, we can replace starvation by caloric restriction mimetics that don't cause any reduction of body weight. So, for instance, hydroxycitrate in combination with chemotherapy, here the red line, gives a better outcome than chemotherapy alone, and hydroxycitrate in this model has practically no effect, at least no statistically significant effect on tumor growth on its alone. And so we can use many different agents that affect the acetyl-CoA depletion that I will not show to replace hydroxycitrate as well. And very similarly, if we use spermidine, the natural component that inhibits EP300 that is in our diet, although at variable levels, you can again show that supplementation with spermidine in drinking water improves the effect of chemotherapy in an immune-dependent fashion. Very similar results can be shown by resveratrol, the activator of sirtuin-1 deacetylase activity. So the bottom line is you can use caloric restriction mimetics to improve anti-cancer immune responses that are favored by chemotherapeutic intervention. And you can show this as well by following in the next phase of the treatment with so-called immune checkpoint blockers, so agents that activate the anti-cancer immune response by taking away the brakes that usually block the process, and show that tumors that already are established are actually cured in most of the mice by a combination of caloric restriction mimetic or fasting plus chemotherapy plus immunotherapy in a precise sequence that we are now optimizing in our laboratory. So I have no time to go into details. It's just a conceptual talk here. A little bit like a biotech company, we are doing two things. We are, on one hand, trying to find the right caloric restriction mimetic for the right indication in our preclinical work. And so to match these optimal combinations, today I've been mentioning the capacity of hydroxycitrate or spermidine or resveratrol or their synthetic, not analogs, but they're synthetic mimetics in the sense that they target the same enzymes as the natural compounds and their capacity to improve the efficacy of chemotherapy, or the combination of chemotherapy with immunotherapy. I've not have time to mention that hydroxycitrate and resveratrol can be used for the prevention of KRAS-induced non-small cell lung cancer and APC-min mutation-induced colon cancer in mouse models. And this again is an immune-dependent and autophagy-dependent pathway. I've mentioned that caloric restriction mimetics in general are allowing to increase lifespan in model organisms, so they are truly non-toxic, because you cannot imagine that an agent that prolongs lifespan would have any kind of toxicity. And we also are working on the utilization of caloric restriction mimetics to reduce weight gain upon high-fat or high-sugar diet and to avoid obesity-related comorbidities like diabetes in mice. And again, for instance, aspirin is working in an autophagy-dependent fashion through deacetylation reactions because it is an inhibitor of EP300 in the same way as spermidine to mediate anti-obesity and anti-diabetic effects. And we speculate that its or cardioprotective anti-arteriosclerosis effects are mediated through autophagy induction as well. And we just published recently 2 clinical studies, phase 2 clinical studies, for the treatment of cystic fibrosis, which is the most frequent lethal monogenetic disease in humans due to the mutation of the CFTR chloride channel, causing what is in French called mucoviscidosis, and in English cystic fibrosis. So this is a disease that affects children and adolescents, leading to respiratory failure. And we could show that a combination of epigallocatechin gallate, which also inhibits EP300, like spermidine and aspirin, plus cysteamine, which is a proteostasis regulator, can actually reverse the loss of function of CFTR in vivo and improve pulmonary inflammation in these children, which is another proof that you can induce autophagy to a clinically meaningful level using caloric restriction mimetics without any major toxicity in humans. The other thing that we are doing is that we are screening for new caloric restriction mimetics among natural compounds. So we use flavonoids and polyphenols that are in our diet, create libraries from them, and look for compounds that might be caloric restriction mimetics based on their effects on mammalian cell cultures, namely absence of toxicity, coupled to induction of autophagy and protein deacetylation. And together with Frank Madeo, we are looking also into direct anti-aging effects in the simplest model organisms that you can imagine, which is Saccharomyces cerevisiae, yeast. So we do chronological lifespan experiments in yeast and look for compounds that prolong longevity to then test them in higher organisms like worms and flies to confirm their effects and to identify potential new anti-aging pro-autophagy compounds. So these are the 2 topics that are dealt in our laboratory. The concept that anti-cancer immune responses are relevant for our final victory over cancer, and the idea that autophagy is important for anti-aging strategies, and knowing that aging is actually the major cause of cancer. Cancer is a comorbidity of the normal aging process. We are trying to establish mechanistic links. So for instance, we know that induction of autophagy in cancer cells can stimulate the immune recognition. And of course, we are looking also for common denominators of these mechanisms that may be situated at whole body metabolism, but also at the level of the microbiome, on which Laurence will telling you a little bit more. So we'll finish here with an outrageous speculation. Maybe health, including the absence of cancer, is just the manifestation of a resilient pluricellular ecosystem that includes the microbiome. So you cannot consider an individual as such. You always have to include the microbial component in the exploration of the entire system, health and disease. And of course, it is very interesting to speculate that caloric restriction mimetics might the microbiome or vice versa. For instance, spermidine, 1/3 of the spermidine we have in our body at least is coming from the bacteria that are producing this polyanion in our gut. I would also suggest, in a very radical way, that cancer is just another comorbidity of aging. And if this is true, then all longevity-enhancing measurements should have an anti-cancer effect, again, with questions on the links to metabolism and microbiome. And finally, I would say that if immunosurveillance is one of the major oncopreventive mechanisms, then most of the longevity-extending regimens should stimulate anti-cancer immunosurveillance, perhaps, again, through an effect on the microbiome. And I will stop with this provocation, and I'm looking for the discussion later on.